Good morning, dear students, or good afternoon, dear students already. We start our lecture, and today we will tell about vitamins. I hope that all of you knows that in additional of fat, carbohydrates, proteins, inorganic salt, oxygen, water, a number of organic compounds are necessary for the normal metabolic processes, for the normal metabolism, as well as for the life growth and health of the body. And these compounds required in very small quantities. They are vital to life processes, and these compounds are called vitamins. Such vitamins can be defined as a group of biomolecules other than fat, carbohydrates, and proteins, most of which cannot be produced by the body and must be ingested with food. But it must be clarified that vitamins themselves are never used for plastic processes and energy production. They have another functions, biochemical functions in our body, and about these functions we will tell with you today. First of all, let's tell a few words about the history of vitamin study. Back in the 7th century, there were separate observations of scientists that a person with a long-term meager and monotonous diet can develop dangerous disease like scurvy, rickets, polyneuritis, night blindness, often fatal. In the second half of the 9th century, scientists had no doubt that symptoms of diseases similar to those of humans were observed in a number of domestic animals. To find out the causes of this dangerous disease, a number of studies have been carried out using various artificially prepared food mixture. And one of the first attempts to feed animals with these mixtures was made by the Russian scientist Nikolai Lunin. In 1881, he showed that prolonged feeding of mice with a mixture of proteins, fat, and carbohydrates extracted from the milk, with the addition of all necessary minerals, salt, and water, led to death of animals, while the control group receiving whole milk developed absolutely normally. On the basis of these experiments, Lunin came to the conclusion that in order to maintain a normal physiological state of the body, some unknown substances which contained in the milk and absence in artificial food mixture are needed. However, this conclusion received general acceptance much later, when these substances, the presence of which Lunin pointed out, were discovered. Only in 1912, the Polish scientist Casimir Funk isolated a substance that cured beriberi from rice bran and called it a vitamin, from Latin vita, life, and amine, because he decided that this chemical group, NH group, is a characteristic feature of these substances. But later, it turned out that amine is not present in all the vitamin found. Therefore, the last letter E was deleted. But till this time, the term vitamin is used. Funk's research led to the foundation for the comprehensive study of vitamin. As a result, vitaminology, a whole study of vitamin, has become a large, rapidly developing branch of knowledge. Science initially chemical nature of vitamin was unknown, and the difference only in the nature of their physiological action. It was proposed to designate vitamin with the letter of Latin alphabet A, B, C, D, E, K. When studying vitamins, it turned out some vitamins, in particular vitamin B, are actually a group of different vitamins which have been designated as B1, B2, B3, and so on. A person needed about 15 vitamins, and mainly we divide them into groups: fat-soluble vitamins, A, D, K, A, and water-soluble vitamins, B complex and vitamin C. Metabolic disorders are often associated either with deficiency of vitamins or the problem with the absorption. But first of all, let's compare fat-soluble vitamins and water-soluble vitamins. They have, you see, different physical property, different solubility, and due to this, they have many different differences between each other. Such way, they absorbed in different manner. Water-soluble directly into the blood without any problem. Fat-soluble only, like all other lipids, inside of micelles, and after that chylomicrons, first into the lymphatic system and only after that into the blood. In the blood, water-soluble vitamins, they travel freely because they are water-soluble. For the transport of fat-soluble vitamin, necessary or a special carrier protein or albumin. Water-soluble vitamins, they can't accumulate in our body. That's why from here necessary to supply them every day. We are needed in frequent doses of this vitamin. You see, and this vitamin very easy excrete from the body with urine. Fat-soluble vitamin can accumulate in our body, can store mainly in liver and adipose tissue. That's why not need to intake them every day, enough one large dose for a week or even months. And they have a problem with excretion, necessary some steps of their transformation, and due to that fact they can be stored in the body, possible toxic effect of them, hypervitaminosis of this vitamin, not only hypo and avitaminosis. Now let's tell about these disorders, metabolic disorders, which are often associated with the or deficiency or excess of this vitamin in the body. If we tell about deficiency, usually we can tell about two situations, or with complete lack of vitamin in food, or with violation of the assimilation and transport. These factors lead to development of avitaminosis, that is, decreases arising from a complete lack of food or complete violation or absorption of certain vitamin. And another situation, hypovitaminosis, caused by the insufficient intake of vitamin from food or their poor absorption, not absolutely. Generally, main reasons of vitamin deficiency can be the next: or reduced consumption, or impaired absorption due to the impaired secretory or intestinal motility in gastrointestinal diseases, or diseases of liver and the pancreas. The same can lead first of all to impaired of fat digestion, and as a result impaired absorption of fat-soluble vitamins. Different metabolic disorders or additional needing in certain physiological or pathological condition, pregnancy, lactation, thyroid toxicose, cachectic diseases, can lead to hypovitaminosis. Antivitamin, the same can be reason of hypervitaminosis. Some antibiotics that suppress the intestinal microflora, because later we will see that some vitamins are synthesized by this intestinal microflora. And we will also discuss pathological condition caused by the excessive intake of vitamin from the food, hypervitaminosis. Today we will tell about water-soluble vitamin, and we start from B complex. These vitamins, they are chemically not related one to another. They have the same functions. All of them, they are coenzymes. See on this picture, you see, its main metabolic waves that we studied with you. Carbohydrates metabolism, amino acids metabolism, fat, lipid metabolism, the center of our metabolism, TCA cycle, and one of the main results of the catabolism of these substances, it's a biological oxidation and oxidative phosphorylation will lead to production of the energy in our body. First vitamin that we will study with you today is vitamin B1, and if you will see on this hymn with attention, all of these our reactions in our metabolic way, they catalyze with the using of different vitamins. That's why they play this very important function in our body. They are coenzymes of enzymes which catalyzes these reactions. Now vitamin B1 or thiamine or anti-beriberi or anti-neuritic vitamins. Recommended dietary allowance of this vitamin 1.2 to 2.2 milligram per day. But the requirement of thiamine depend from caloric content of the diet. If the carbohydrates content of the diet is excessive, the increase of thiamine intake will be required. Here you can see the richest sources of vitamin B1 include yeast and animal liver, the richest source of this vitamin, but pork, organ meat, green leafy vegetables, legumes, sweet corn, egg yolk, cornmeal, brown rice, jam, and husk of the grains, berries, and nuts are other rich sources of this vitamin. As all other water-soluble vitamins, vitamin B1, the same is not stored in the body and must be supplied daily. Structure of this vitamin. Vitamin B1, first of all, I want to repeat again that it is the first vitamin isolated in crystalline form by Casimir Funk, and thiamine is derivative of substituent pyrimidine ring and thiazole ring, which are linked by the methylene bridge. Thiamine is only natural compounds with thiazole ring. Thiamine in this form of vitamin B1 is absorbed from the small intestine, but thiamine is not active vitamin. That's why thiamine is rapidly converted to its active form, thiamine pyrophosphate. You see, we attach to this position 2-phosphate group by the enzyme thiamine pyrophosphokinases 1. And here you can see the active form of this vitamin, coenzyme thiamine pyrophosphate, TPP. TPP is required as a cofactor for many reactions, But first of all, for three very important dehydrogenases. These enzymes are a complex of pyruvate and alpha-ketoglutarate dehydrogenases, both of which are associated with TCA cycle and energy production. Not so far ago we discussed branched-chain keto acid dehydrogenases required for the metabolism of branched-chain amino acid like leucine, isoleucine, and valine, and we tell that disorder, deficiency of this enzyme, lack of this enzyme lead to maple syrup disease. These three dehydrogenases also required another cofactors, derivative of vitamin lipoic acid, pantothenic acid, CoA, riboflavin, and niacin. And we will talk about them later. I want to tell about this now again, and we will use this reaction to show the role of these vitamins many times. The lack of thiamine leads to serious decreases in ability of cell to produce energy and carry out reductive biosynthetic reaction due to its role in these three most important complexes of dehydrogenases. In addition to this, thiamine pyrophosphate is a necessary cofactor for transketolase, catalyze reactions of the pentose phosphate pathway. Here again, I show you full scheme of this vitamin using, so it's a cofactor for pyruvate dehydrogenase complex. Now I hope that you recognize it. Do you remember, after glycolysis or pyruvate, it's possible to produce from some amino acid. Pyruvate, under the action of this complex, pyruvate dehydrogenases with three enzymes, with 5 coenzymes, and one of these coenzymes is thiamine pyrophosphate, is converted to acetyl-CoA after its oxidation and decarboxylation. The same reaction we can see in TCA cycle when alpha-ketoglutarate is transformed in succinyl coenzyme A. Absolutely the same mechanism we can see in catabolism of branched-chain amino acids, and it's the same mechanism in these three variants, three dehydrogenases. And one more role of thiamine is it is a coenzyme of transketolases and pentose phosphate pathway. How you will answer on this question about different vitamins during our classes and on exam? Of course, necessary to draw the structure. It is necessary to tell about the activation, about the active form of this vitamin. After that, the main biochemical role of this vitamin in our body, and after that, as example, you have to write any one reaction with this vitamin as a coenzyme. This reaction, formation of acetyl-CoA, or the same, you can write formation of succinyl-CoA, is very useful in this case for you, because as I already tell, here are 5 coenzymes, 4 of them vitamins. That's why you can use this reaction as minimum 4 types for demonstration of the biochemical role of 4 different vitamins. Now we will use it for explaining of the role of thiamine pyrophosphate in our metabolism. It's a coenzyme, one of the coenzyme in this reaction. Here directly I show you the role of our thiamine pyrophosphate in this reaction. It starts this reaction, because first of all happened the attachment of our pyruvate after its decarboxylation to first enzyme, pyruvate dehydrogenase, but correctly named this enzyme pyruvate decarboxylase, but traditionally we tell pyruvate dehydrogenase with thiamine pyrophosphate like coenzyme. Absolutely the same mechanism, only instead pyruvate here, alpha ketoglutarate. Instead acetyl-CoA, succinyl coenzyme A. But mechanism the same, and again we show the role of thiamine pyrophosphate. You can use both of this variant. Pentose phosphate pathway. I hope that you remember from previous semester that it's an alternative variant of glucose oxidation. Why alternative? Because we don't produce energy in this pathway, and we produce two another very important substances like NAD, pH, and ribose 5-phosphate. Each of them, they have a special function in our body. We produce them during the first oxidative phase of pentose phosphate pathway. But we have second stage, non-oxidative stage, and during this stage, excess of pentoses can be converted due to a series of different reactions into triosis and hexosis. Glyceraldehyde 3-phosphate and fructose 6-phosphate, which after that can be used in glycolysis for energy production. And one of the type of reaction here is transketolysis. There are two types of reaction, transketolysis and transaldolasis. But only transketolysis here and here, they have as a coenzyme vitamin B1, thiamine pyrophosphate, or thiamine diphosphate in some books. Now let's talk about symptoms of this vitamin deficiency. The lack of thiamine effectively in the nervous system and circulation, but in stomach and intestine the same. The earliest symptoms of thiamine deficiency includes constipation, appetite suppression, nausea. Progressive deficiency will lead to swelling of nerves, increased pulse rate, difficult breathing, dyspnea, motility, dyspepsia, weakness, and heart irregularities. Mental depression, peripheral nephropathies, and fatigue. Chronic thiamine deficiency lead to more severe neurological symptoms, including ataxia, mental confusion, loss of eye coordination, nystagmus, especially vertical nystagmus. It's a characteristic symptoms of thiamine deficiency. Cybele cause very dangerous disease. There are two clinical symptoms of beriberi: polynephritic or dry, and edematous or wet. Polynephritic or dry beriberi, in this condition, CNS manifestations are the major features. Muscles becomes progressive wasted, walking becomes difficult, peripheral neuritis with sensory disturbance lead to complete paralysis. Edematous or wet. Here already the cardiovascular system is prevalently affected. So, on the first place, manifestation of cardiovascular system disorders. Here we can see edema of legs, face, trunks, serous cavities. They are the main features. Palpitation, breathlessness, cardiomegaly, increase the size of the heart, resulting in cardiomyopathy. Death occurs due to the heart failure. Possible infantile beriberi. If the mother has a thiamine deficiency and her milk is poor of this vitamin, then the baby also develops beriberi symptoms, such as vomiting, convulsion, sleeplessness, restlessness, cardiac dilatation. Additional thiamine deficiency related syndrome is known as a Wernicke-Korsakov syndrome, which is most often associated with chronic alcohol consumption. This disease is most commonly found in chronic alcoholics due to the fact that, first of all, alcohol impairs thiamine uptake from the small intestine, as well as the fact that these individuals generally have poor dietetic lifestyles. It is characterized by loss of memory, apathy, and nystagmus. Without treatment lead to coma and death, of course. All these symptoms appear signs as a result of thiamine B deficiency. The activity of dehydrogenases of keto acid decreases. It leads to accumulation of these keto acids, pyruvate, alpha-ketoglutarate, and so on, in the blood, and then in the brain, and it is very harmful. The activity of transketolysis also decreases, especially in erythrocytes. And the deficiency of thiamine pyrophosphate also leads to impairment in nerve impulse. But on the first place, of course, problem with energy production. Now next, let's tell about vitamin B2, second vitamin for today, or riboflavin. Recommended dietary allowance of this vitamin, 2 to 4 milligrams. Here you can see foods which are rich of this vitamin. Common sources are organ meats, milk, cheese, egg, green leafy vegetables, meat, whole grains, and peas. I hope that you have a breakfast today and that you are not angry, because this picture are very appetite. Now structures, and you have to know the structures, not of this vitamin. See here, the chemical basis of riboflavin is isoalloxazine, heterocyclic three-ring structure, which attached to D-ribitol through the nitrogen atoms. Ribitol can be obtained by reducing ribose sugar. See, I hope that you recognize it. It's a alcohol, of course, and this alcohol can be produced from ribose due to its reduction. Riboflavin is a yellow crystal that is thermally stable but sensitive to light. Under the influence of ultraviolet rays, it turned into lumiflavin, which exhibits yellow fluorescence. You will see this experiment during our classes. But riboflavin itself is the same as inactive. It's only precursor of active coenzymes, flavin mononucleotide. If we stop here, this is flavin mononucleotide, or flavin adenine dinucleotide, FAD. FAD is more popular in our metabolism, but FMN the same plays its role. These flavoenzymes, they catalyze, first of all, the oxidation and reduction of different substrates. See on this picture, to this nitrogen, they play this nitrogen in isoalloxazine ring. They can be acceptor of 2 electrons and 2 protons. And due to this ability, we can transform our FAD or FMN into FADH2 or FMNH2. So they may be like donor, such acceptors of two hydrogen atoms, 2 electrons and 2 protons. Due to this ability, the coenzymes FAD and FMN are associated with certain enzymes involved in carbohydrates, lipid, proteins, purine metabolism, besides the electron transport chain, because they can take part in oxidation and reduction. But here we can tell about two types of reaction with FAD and FMN like coenzymes. First type of reaction with participation of oxygen. In this case, enzymes, they are oxidases. For example, amino acids, oxidases, we took part in oxidation of amino acid. One of example of amino acid deamination, if you remember. And second part of reaction, dehydrogenases, attachment or removal of electrons and protons. These types play the major role in biological oxidation and other type of oxidation. For example, they participate in reaction that we consider above. Pyruvate dehydrogenase, alpha-ketoglutarate dehydrogenase complexes, the brain chain alpha-ketoacid dehydrogenases, additional important metabolic regulatory enzyme that require flavin as a coenzyme include succinate dehydrogenase in TCA cycle, and it's I will show you later in a complex number 2 of biological oxidation. Acyl coenzyme A dehydrogenases in beta oxidation of fatty acid. Glycerol 3-phosphate dehydrogenases involved in glycerol phosphate shuttle and triacylglycerol synthesis. Xanthine oxidase. We started xanthine oxidase 2 weeks ago. Yes, involved in the purine nucleotide catabolism. So a lot of example of the using of this vitamin like coenzyme. Here you see in TCA cycle, one time we can see this vitamin in alpha-ketoglutarate complex, because as you remember we tell that there are 4 vitamins, B1, B2, B3 and B5. And B2 the same plays its special role in this complex. After that, we have one more reaction. Which will require succinyl coenzyme A dehydrogenase, which we used for succinate dehydrogenase, which used vitamin B2, FAD, as a coenzyme. Here I show you this reaction with structures of succinate and fumarate. This example in Krebs cycle possible you see in beta oxidation. Not only NAD but FAD depend dehydrogenase, acyl coenzyme dehydrogenase, the start of this process. Here you can see this reaction bigger. You see, when we use fatty acid, active fatty acid for energy production, and first of all we use this B2 vitamin, riboflavin, for this reaction. That's why it's necessary for the and carbohydrate and lipid, and as I tell earlier about in amino acids metabolism. If we use xanthine oxidase, it's necessary for the DNA metabolism. And pay attention that this FADH2 after that carry his electrons and proton to electron transport chain for the production of energy. And you remember that from one FADH2 it is possible to produce 2 molecules of ATP. And succinate dehydrogenase complex, it's a part of the second complex in our respiratory chain. the same very important function of this vitamin. Deficiency symptoms of this, the lack of this vitamin include itchy and burning eyes, angular stomatitis, halosis. What is halosis? It's cracks in the corner of the mouth. I will show you picture later. Bloodshot eyes, glossitis, inflammation of the tongue, seborrhea, tremors, lethargy, photophobia, dizziness, inability to urinate. Here are my symptoms. Here the picture, you see glossitis. After that, dermatitis, stomatitis, symptoms of this vitamin deficiency. Really, these symptoms, especially dermatitis, it's a common symptoms for almost all vitamin deficiency. After that, chalosis, you see inflammation, cracks in the corner of the mouth, seborrhea. That's all. Okay. Now let's tell about one more very interesting vitamin. Why interesting? Because here usually we confuse. Because in our Russian classification, this vitamin is vitamin B3. But in your international classification, in your book, Pantothenic acid is vitamin B5. And usually you confuse, but please remember that if in our task, in our question, we write vitamin B3, it means that we ask you about pantothenic acid. Now, recommended dietary allowance of this vitamin 3 to 10 milligrams. Pay attention on the structure, but full structure I will show you later. It's rather complex, that's why not need the study to study the structure of this vitamin. Distribution widely. From here the name of this vitamin, because pantos from Greek means everywhere, present in all type of food. And eggs and liver and yolks and yeast and green vegetables, always you see, and can be synthesized by intestinal microflora. That's why, of course, deficiency very rare. We can see deficiency of this vitamin. What is the function of this vitamin in our body? First of all, structure. This vitamin consists from these three components, and one of the component is this pantothenic acid. Here you see 3-5 ADPE, very interesting, present here, and mercapethylamine structure with the sulfur group. This sulfur is very important for us. That's why we see as coenzyme A usually, as coenzyme A, because pantothenic acid is a precursor of active form of coenzyme A. Is a component, basic component of coenzyme A. And I hope that you know, because we every time use this coenzyme A for activation of different substances, because coenzyme A functions in the transfer of acyl group with the formation of T-ester bond in acyl coenzyme A, and this bond is a high energy bond. Pay attention on this. And where we can see this coenzyme A, in acetyl coenzyme A, in acyl coenzyme A, in succinyl coenzyme A. Always when you see S-sulfur coenzyme A, you already have to understand that B3 vitamin, Pantothenic acid, Pantothenate, took part in these processes. Due to this, coenzyme A is a center molecule of our metabolism. You see, it's necessary, and for a little later I will show you a scheme and we tell about this. First of all, let's tell about these reactions again. I hope that now, even you don't know this reaction before this, but already now you have to recognize this reaction, because I repeat this reaction already third time and will repeat one more time in next hour step. You see, it's again formation of acetyl-CoA from pyruvate, and together with vitamin B1 and B2, B3 vitamin, Pantothenic acid, the same took part in this reaction. It's a second coenzyme or second enzyme in this pyruvate dehydrogenase complex. See here that acetyl-CoA is a central molecule of carbohydrates, lipids, and protein metabolism. And carbohydrates, and fatty acid, and proteins after catabolic pathway, they are converted to acetyl-CoA. And such way, Pantothenic acid, we can tell about Pantothenic acid, that it may be regarded as a coenzyme of metabolic integration, because it integrates all these directions of our metabolism. Moreover, Pantothenic acid is not only took part in catabolism, it took part in biosynthetic processes of higher fatty acid, neutral fats, ketone bodies, steroid hormones, hemoglobin, acetylcholine, and many, many other. So that's why Pantothenic acid plays very, very important role in our body. But for example, here in biosynthesis of fatty acid, we used acetyl-CoA as a start of synthesis, and malonyl-CoA, if you remember, after condensation of them, we start to produce fatty acid. But one of the additional role of Pantothenic acid, it's not only the part of coenzyme A. If you remember that fatty acid synthesis, I think that you remember that very complex enzyme, fatty acid synthase, one of the men has ACP, acyl carrier protein. It's one of the men of fatty acid synthase. And Pantothenic acid serves as a component of this acyl carrier protein. That's why we can see Pantothenic acid not only in coenzyme A, but in ACP the same. Deficiency of this vitamin is extremely rare due to its wide distribution in all parts of food. And symptoms associated with Pantothenate deficiency are difficult to assess because they are resemblance those of other vitamin deficiency. They are absolutely the same as in other vitamin deficiency. What we can see here? As usually, dermatitis, lesions of muscles membrane, dystrophic alterations of endocrine glands and nerve system, cardiac and renal alteration, Here depigmentation, and not only depigmentation, but alopecia, local loss of the hair, growth retardation, loss of appetite. These are the main symptoms of this vitamin deficiency. But when we can see deficiency of this vitamin? Only if we have diet which are poor of vitamins. But it is very difficult to prepare any food with deficiency of only vitamin B3. Usually, if we see deficiency of B3 in our life, it's caused together with deficiency of other vitamins. And these symptoms that we describe here, it's usually a result of experiment with different animals, when we give these animals a food with poor of only this vitamin. So it's experimental symptoms. Next vitamin that we will discuss today, niacin, or nicotinic acid, or nicotinamide, or PP vitamin, Pellagra preventive factor. You see how many names, or common name of nicotinic acid and nicotinamide, niacin, or B3 vitamin in international classification, and pay attention, in our Russian classification, this vitamin is B5, I confuse you more, B5 vitamin. That's why in our questions or task or MCQs, we usually always to show you, if we tell about this vitamin, we usually use name PP vitamin, Pellagra preventive factors. But remember about this classification. If we ask you B5 vitamin, we ask about niacin. Recommended dietary allowance of this vitamin, 15-25 milligrams, rather high amount, quantity, because this vitamin is very important. It's a top of coenzymes in our body. I will show you later this. Distribution of this vitamin. Rich sources of niacin usually are meats, poultry, fish, liver, kidney, eggs, nuts, peanuts, butter, yeast, wheat germ, and pay attention that part of PP vitamin can be obtained from tryptophan. Really, niacin is not true vitamin in the strictest definition. Science NAD can be derived from the amino acid tryptophan, and we discuss it when we tell about amino acids. However, the ability to utilize tryptophan for NAD synthesis is inefficient, given that approximately 60 milligrams of tryptophan are required to synthesis 1 milligram of NAD. Also, synthesis of NAD from tryptophan required another vitamin, B1, B2, and first of all, B6 vitamin. You see that tryptophan cannot totally replace niacin due to this. Only about 10% of active coenzymes, active NAD, plus can be synthesized from tryptophan. That's why it's in all time of our life, it is necessary, require this vitamin from food. Niacin itself, in this case nicotinamide, it is inactive precursor of active coenzymes NAD or NADP if here a pair phosphate group. So we have two active forms of these coenzymes, NAD plus and NADP plus, and approximately 200 of enzymes, primary dehydrogenases of course, require these coenzymes because they act as a hydrogen or electron donor or acceptor. And pay attention that here we can attach both electrons but only one proton. One electron, due to reduction, is attached to this nitrogen, and one electron and proton to this carbon. See, and NAD plus is converted to NADH plus H plus because second hydrogen continue be in environment. The same about NADP. So NAD and NADP, they are differs only due to phosphate group here. In all other part of their formula, they are absolutely the same. But it's only they are similar one to each other, but they have different functions in our body, and they catalyze different reactions. If we tell about biochemical functions of this reaction, first of all, you see that they took part in oxidation and reduction. They are coenzymes mainly of dehydrogenases. And here we have a number very high, more than 500 of different reactions. They used niacin as a coenzyme. I want to show you another one scheme. See on this scheme. See, it's a main scheme of our metabolism. Is with green color, we show you the role of NAD and NADP in our body. See, first of all, again, NAD took part in keto acid oxidative decarboxylation reaction in pyruvate dehydrogenase and alpha ketoglutarate dehydrogenase complex. But in TCA cycle, we have two more reactions with production of NAD. And here you see, and here in malate dehydrogenase, the same has NADH as a coenzyme. If you remember, in anaerobic glycolysis, we use lactate dehydrogenase with NADH as a coenzyme. If we tell about glyceraldehyde metabolism, here the same, we use NADH in glycolysis itself, in beta oxidation, third reaction of beta oxidation with NAD. We used NAD dehydrogenase. So very, very many. The same we can tell about amino acids metabolism, about some amino acid synthesis and oxidation of ethanol and many. It's a major role. It's a chief of all our coenzymes, but one of the in our metabolism. But the major role of NADH is to transfer of its electrons from metabolic intermediates to electron transport chain, thereby producing adenosine triphosphate, ATP. So this enzyme plays a very important role in energy production. Remember. NADPH serves as a cofactor for a number reducing biosynthetic processes, including fatty acid synthesis, cholesterol biosynthesis, steroid hormone biosynthesis, bile acid synthesis, oxidation of glutamate, I will show you, synthesis of deoxyribonucleotides, that after that necessary for DNA synthesis, but never for energy production. The lovely hour reactions. And NAD here is the last fourth vitamin in this reaction. You see that if you know this reaction, you know answer on the question about 4 different vitamins, B1, B2, B3, and at last niacin, B5 vitamin. Even I already tired from this reaction, show you 4 times already. I hope that known reaction for you, because we started it in this semester when we tell about deamination of amino acid and the famous, the most activity enzymes in our amino acid deamination is glutamate deaminase, and this glutamate dehydrogenase, and this enzyme has, or sometimes NAD, it depends from the direction, deamination or reamination, or NADP as a coenzyme, non-protein part of this enzyme. This reaction I'm sure that you know. If we tell about the deficiency is so interesting that we have even a special name of this. It's already not symptoms, number of symptoms, but syndrome. And the name of this syndrome is Pellagra. It's a disease of normally 3D, dermatitis, diarrhea, dementia. Without treatment, a pair of false Ds, it's a pity, death. If we tell about dermatitis, the skin becomes very, very rough, and it gives the name of this vitamin and this disease, because in Italian, pellagra means rough skin. That's why the name of this disease, pellagra, and one of the main of vitamin, pellagra preventive factor. And pay attention that this rough skin, especially in sun-exposed area of the skin, not all, but only open sun-exposed area. Together with these 3D, sometimes appear another general symptoms like stomatitis, gingivitis, psychosis, psychoneurosis, hallucination, muscular weakness, general tiredness, loss of appetite, irritability, nausea, vomiting, tendons, gums, tension, depression. The same may be present, but first of all, it's a disease of 3D. One more vitamin for today, vitamin B6. This vitamin, I hope, will be not difficult for you in this semester, because the main role of this vitamin we discussed with you many times. But first of all, let's tell about vitamin. There are 3 forms of this vitamin: pyridoxine, pyridoxal, or pyridoxamine. But in all of them, they are inactive. If we tell about recommended daily requirement for this vitamin, it's about 2 milligrams per day. Distribution is the same, rather wide. Wheat, corn, peas, beans, potatoes, egg yolk, liver, muscle meat, whole grains, soybeans, peanut, many other fruits and vegetables, and yeast, milk and green vegetables provide less. They are more poor of this vitamin. Part of this vitamin can be synthesized by the intestinal microflora the same. Now let's see the structure of this vitamin. You see, maybe three-fold. Pyridoxine is the pen of the group from here in this position. If alcohol, pyridoxine. If aldehyde, pyridoxal. If amine group, pyridoxamine. But all of them, they are inactive. And all of them, they are precursor of active, biological active form of this vitamin, pyridoxal phosphate, after phosphorylation of this carbon. A special enzyme, pyridoxal kinase, with ATP took part in activation of this vitamin. And it's a pity, but you have to know the structure of this vitamin the same. So it's not difficult. Usually you know this vitamin. Pyridoxal phosphate acts as a cofactor in all enzymes that carry out the transamination, aminotransferases. And we discussed this reaction with you today. So all aminotransferases have this vitamin as a coenzyme. These reactions, you know, they require and for the catabolism of amino acids and indirect elimination of amino acids and for biosynthesis of amino acids, because these reactions, they are reversible. Pyridoxal phosphate is a cofactor for synthesis of some neurotransmitters, including serotonin, catecholamines, histamine, acetylcholine, GABA, and others, because this coenzyme, this vitamin is a coenzyme of one more very important reactions with amino acid, decarboxylation of amino acids. Pyridoxal phosphate is required for the synthesis of heme through the first condensation reaction. A special enzyme, aminolevulinic acid synthase, has this vitamin as a coenzyme. And later I will show you this reaction of condensation when glycine interact with succinyl coenzyme A, and we produce first product of heme synthesis, aminolevulinic acid. Pyridoxal phosphate is a cofactor for two enzymes involved in the metabolism of methionine and cysteine. As discussed above, pyridoxal phosphate is also required for the condensation of tryptophan to NAD. Pyridoxal phosphate is also required for the glycogen homeostasis, because it's a cofactor for glycogen phosphorylase. So more than 100 of different enzymes they have B6 vitamin as a coenzyme. So it's on the second place in the popularity in our body, like coenzyme. Now let's see on a few examples of these reactions. First of all, transamination. Transamination, I'm sure that you know even better than I. And you see that pyridoxal phosphate play the center role in this ping-pong reaction, because this amino group, it is transfer not directly to the products, but through the active site of this enzyme, and first of all, it's attached to our vitamin, to pyridoxal phosphate. Decarboxylation of all amino acid with the production of biogenic amines, in this picture production of GABA from glutamate, the same we use pyridoxal phosphate. I tell you about them, all of this reaction. Now I show you this reaction. One of this reaction you can use for demonstration of biochemical function of this vitamin on exam. heme synthesis. This reaction I only show you, not need to study, because we don't study reactions of heme synthesis, but I tell about this reaction. That's why I show you this reaction. When glycine interact with succinyl coenzyme A and B produce aminolevulinic acid. And again, pyridoxal phosphate is a coenzyme of aminolevulinic acid synthase. If we tell about vitamin B6 deficiency, very rare, because it's present in all parts of our food and can be synthesized in microflora. Usually taking of isoniazid, a drug often used for treatment of tuberculosis, can lead to deficiency of this vitamin because it forms inactive derivative, inactive complex with pyridoxal phosphate. What is the main symptoms of B6 deficiency? Pimples around the eyes, nose, mouth, the behinds of the ears, chapped lips, nervousness. Violation of the synthesis of it's a result of disorder of biosynthesis of biogenic aminos, epinephrine, norepinephrine, GABA, serotonin. It leads to disorder of the nerve system, problem with the nerve system. Insomnia, muscle weakness, pellagra-like dermatitis, because we remember that this vitamin necessary for NAD synthesis, cramps in the arms and legs, hair loss, anemia. That's why the main symptoms of this vitamin deficiency. Usually this vitamin is your lovely vitamin in this class, this topic. And this is my lovely vitamin from all. It's a vitamin H or biotin or vitamin B7. You see how many names. And only biotin is sulfur anatone. It's one of the sulfur-containing complex of B vitamins. Here you see present sulfur. This vitamin, when separated, it's a colorless, crystalline, and of course water-soluble vitamin. And recommended dietary dose of this vitamin 0.25 milligrams. Structure, it's simple really, but sometimes you confuse when you draw it. And now I want to repeat again what structures you have to study. You have to know structure of vitamin B1, of vitamin B2, of PP vitamin, of vitamin B6, biotin, and finally, today, after the lecture, the final vitamin that we will discuss today, vitamin C. So you have to know the structure of 6 vitamins. Not need to study the structure of vitamin of pantothenic acid, B9, and B12, because they are complex. Distribution. Biotin present in almost all food products. Here especially the must-rich products with this vitamin: liver, milk, egg yolk, kidney, brown rice, yeast, peanut, cauliflower, fungi. And this vitamin, biotin, the same, can be produced by intestinal bacterials. I already tell about RDA of this vitamin, 0.3 milligrams. About 0.3, 0.25, 0.35 milligrams. And why it's my lovely vitamin? Because very easy to tell about the biochemical function of biotin. It is a coenzyme of only carboxylation and transcarboxylation reaction, in which it serves as a carrier of activated carbon dioxide. And due to this reaction, biotin is involved in the synthesis of higher fatty acid because it's a coenzyme of acetyl coenzyme A carboxylase, glucose synthesis in gluconeogenesis. You know that the main enzyme in this pathway is pyruvate carboxylase. It is a coenzyme of propionyl coenzyme carboxylase, which is convert propionyl coenzyme A into methyl malonyl coenzyme A. We studied this reaction when we learned beta oxidation of fatty acid with odd number carbons atoms and oxidation of valine, methionine, isoleucine, and threonine. Pay attention to this last reaction, because we will see this reaction later when another vitamin, B12 vitamin, this vitamin also involved in this metabolic pathway. So biotin is necessary for biosynthesis of and metabolism of carbohydrates, lipids, proteins, purine nucleotides, and if purine nucleotide, of course, nucleic acid. Let's see why some of these reactions. First of all, if we start to produce fatty acids, first of all, necessary to produce malonyl coenzyme A from acetyl coenzyme A, and we do it with carboxylation. And acetyl-CoA carboxylase has biotin as a coenzyme. Absolutely the same, it's the first reaction you see of gluconeogenesis, production of oxaloacetate due to carboxylation of pyruvate. Pay attention, what is a coenzyme here? Yes, it's our vitamin H, our biotin. For carboxylation also always necessary energy, always. So always we use ATP. And if we tell carboxylation of pyruvate, you remember that one of carbon in the pyruvate is from the carbon dioxide. A little later I will show you this picture and tell about the role of biotin one more time. Deficiency, the same, rarely, because when we can see deficiency of biotin, it may be any absolutely exotic diet can be this diet is rich of raw egg whites, because avidin that present in raw egg whites is an antivitamin of biotin. Avidin binds with biotin and form stable inactive complex. Here may be reason of deficiency, or severe intestinal dysbiosis, for example, after taking a large doses of antibiotics, because a part of this vitamin are produced by the intestinal microflora. Symptoms, usually symptoms of hypovitaminosis: dermatitis, glossitis, loss of appetite, nausea, muscle pain, hallucination, hair loss, mental depression, not specific. I know that you are tired, but try to listen me, because 3 yet vitamins and we will finish for today. And our next vitamin is folic acid, or folate, or vitamin B9, or vitamin B6. Folic acid was isolated in 1941 from green leaves of plant, and in connection of it we have this name, folic acid, because from Latin folium, leaf. FDA recommended dietary allowance 0.2 to 0.5 milligrams. But in some situation in the women during the pregnancy or lactation, we recommend higher dose of this vitamin, about 0.6 milligrams. Distribution: mainly green leafy vegetables, liver kidney, lima beans, and whole grain cereals. Folic acid, the same, can be made by the bacteria in the intestine. Avocado, I like to see here. Now let's tell about the structure of folic acid. Folic acid consists of 3 structural units. Pyridine residues, 1. Parabenzoic acid, and known for you, glutamate, glutamic acid. And pay attention that here for us important nitrogen number 5 and nitrogen number 10, because they, through this nitrogen, play important biochemical function in this vitamin. And what is the main function? It's a transportation receives of 1 carbon fragments, but about this later. See on 3 this part: pyridine, parabenzoic acid, and glutamic acid. But folic acid is inactive, and for its activation necessary to reduce it, necessary to attach 4 protons and 4 electrons, and NADPH took part in activation of folic acid. Pay attention, this is inactive, see on this part, and active when we attach 4 hydrogen, 1, 2, 3, 4. 4 hydrogen atoms are attached. That's why the name of active form of this vitamin, tetrahydrofolate. Only tetrahydrofolate can take part in transportation of 1 carbon units, and such way took part in biosynthesis of all of these very important substances in our body. I tell already about the special role of N5 and N10, because we attach 1 carbon fragments such as formyl or formimino or methinyl or methylene or methyl to this carbon, nitrogen. Sometimes only to 5, you see, and due to this we write N5 methyl tetrahydrofolate. I explain you why we use this writing, because we show what group and position of this group in our vitamin. Sometimes we carry this 1 carbon fragment to 10 nitrogen number 10, and we write N10, 10, formyl tetrahydrofolate. Sometimes this 1 carbon fragment, they are attached together, and to 5 carbons, 5th carbons, and 10th carbons, and we write it N5 and 10, methylene tetrahydrofolate, or like this, methynyl tetrahydrofolate. That's why when we see these interesting letters and number, now I think that you understand about what you have to think. It's a what group and position of this group in this vitamin. And you see that this vitamin carries these different 1 carbon fragments from donor to acceptor. What is a donor usually? Donor usually some amino acid, like serine, or glycine, or histidine, and we transfer one carbon fragments from them to different intermediates. And finally, we use these one carbon fragments for the production of important final products, important substances like purines. If you remember, and I show you later that carbon number 2 and carbon number 8 in purines, we take from tetrahydrofolate. And purine is a precursor of nucleic acid, DNA and RNA. Pyrimidine, the same nucleotide, deoxytimidylic acid, D-timid P, the same is required for folic acid, and the same is necessary for DNA synthesis. Some amino acids are produced with this mechanism. Vitamin B9 together with B12 vitamin, later I will show you, plays very important role in methionine homocysteine metabolism. And methionine, initiation of protein biosynthesis in prokaryotic cells, the same are formed with tetrahydrofolate because we add 4-mil, one carbon fragment. You see, remember that we tell that these two carbons, number 2 and number 8, we take from tetrahydrofolate, and 10 or and 5-10, here methionyl, here 4-mil, but it's only the name of these one carbon fragments. But both of them, they are incorporated in our purine. You see here. And that's why this vitamin, folic acid, plays the important role in DNA and RNA synthesis. And I promise to show you biotin role here. You see carboxylation with enzyme carboxylase, and this reaction is catalyzed by enzyme coenzyme biotin. Deficiency of vitamin of folic acid. Usually we can see deficiency of folic acid in situation when the using of folic acid is increased in pregnancy, infancy, in stress, and in this case can appear symptoms of this vitamin deficiency. Deficiency may be result of defective absorption plus dietary deficiency. It may be pathology of small intestine. It may be result of alcoholism, or the same, you have to remember about this, treatment with the drugs that are dehydrofolate reductase inhibitors. So, like methotrexate. Methotrexate inhibits activation of folic acid. And of course, it leads to deficiency because even folic acid in the food is present, but it can't be activated. We can't produce tetrahydrofolate. And usually deficiency of this vitamin leads to growth failure, gray hair, mouth and tongue swelling, diarrhea, and pay attention, megaloblastic anemia. Megaloblastic anemia is developed as a result of diminished DNA synthesis and erythropoietic stem cell. And what else I want to show you, that the lack of folate during the pregnancy lead to neutral tube defect in the fetus. These even have a special name, spina bifida. That's why during the pregnancy necessary to intake this vitamin additionally to prevent this situation. Now B12 vitamins, or cobalamin, or anti-anemic vitamins, or anti-pernicious anemia factors. This vitamin, B12 vitamin, was isolated from the liver in rats in the form of red crystals in 1948, and only in 1955 Dorothy Hodgkin described the structure of this vitamin, because this structure is very complex. And for this work in 1964, she was awarded the Nobel Prize. Vitamin B12, is composed of a complex of tetrapyrrole rings. Structure, the pyrrole rings, you see, and cobalt ion in the center. Neither animals nor plant are able to synthesize vitamin B12. That's why this vitamin, only microorganism can synthesize this vitamin. That's why this vitamin is not present in the plant. It's present only in animals' food. Liver, kidney, fish, whole milk, egg, oysters, fresh shrimp, pork, chicken. And the recommended daily intake of this vitamin, RDA of this vitamin, very, very low, 0.003 milligram, 3 microgram only. See. There are two different active form of this vitamin. It depends what radical is attached here. If it's a methyl group, we tell about methylcobalamin. If it's deoxyadenosyl radical, we tell about deoxyadenosylcobalamin. Two active coenzyme form of this vitamin. And they have different localization and different function. Methylcobalamin localizes in the cytoplasm. Deoxyadenosylcobalamin is localized in the mitochondria. Distribution I already tell, present only in animal products. Milk, cheese, egg, sea products, but not in the plant. Here, very important, the absorption of this vitamin in the intestine, because dietary B12 reaches the small intestine where it, with the action of additional transport proteins, and in the intestine, it is attached with the intrinsic factors. Intrinsic factors is a glycoprotein synthesized by the lining cells of the stomach. And this complex, B12 and intrinsic factors, is absorbed into the cell of the mucous membrane of the terminal ileum. After that, the protein part, this intrinsic factors, protein part, is dissociated and digested in the mucosal cells, and free B12 binds with transcobalamin, and only after that travels to the bloodstream. And one of the factors of this vitamin deficiency is the lack of intrinsic factor. I already tell about two active coenzymes form and two different types of reaction. Such way, methylcobalamin in the cytoplasm involved in the transmethylation synthesis of methionine from homocysteine. The cytoplasmic methionine synthase catalyze the transfer of methyl group from N-methyl tetrahydrofolate, our B-active B9, to homocysteine to yield methionine and to liberate tetrahydrofolate. Such way, our methyl cobalamin takes part in the reactivation of folic acid required later for the synthesis of nucleic acid. nucleic acid precursors of DNA and RNA. Such way indirectly, but methylcobalamin took part in regulation of nucleic acid synthesis. So deficiency of this vitamin lead to disorder of nucleic acid synthesis. First of all, in tissue with a very high intensive metabolism and cell proliferation, and first of all, an erythropoietic stem. If we tell about mitochondrial methylcobalamin, it's a coenzyme of methyl-malonyl coenzyme A mutase, which took part in transformation of methyl-malonyl coenzyme A into succinyl coenzyme A. And we see this reaction when we tell about catabolism of amino acid with odd number carbons atoms and certain amino acid, valine, isoleucine, methionine, and threonine. And later, this succinyl coenzyme A can be utilized in TCA cycle. And when here we tell about this vitamin deficiency in this already type of reaction, it leads to accumulation of intermediate of this reaction. Correctly tell about the substrate of this reaction, methylmalonate. And this methylmalonate is very toxic for the nerve system. Now let's see this reaction. It's a reaction of methylcobalamin. You see, when this I showed this slide to you when we tell about metabolism of amino acid and their role in our body, and when we tell about the role of methionine in our body as a donor of one carbon fragments. And you know that due to this, when these one carbon fragments we use for methylation of different substances, we transform methyl acidanosine methionine, active methionine, into acidanosine homocysteine. And possible reactivation of after that of these through homocysteine production, and homocysteine again can be converted to methionine. And here in this reaction, a very important role plays tetrahydrofolate as a donor of this one carbon fragment. It's transfer, you see, one carbon fragment to, but not for reactivation of this. We use B12 vitamin. Reactivation of our tetrahydrofolate. That's why vitamin B12, cobalamin, the same, took part in this reaction. See, it's a scheme of this reaction, not reactions, but the scheme. When tetrahydrofolate carry this methyl group, after that B12 take this methyl group and use it for biosynthesis of different substances, and our for reactivation of methionine from homocysteine, and our already again active tetrahydrofolate can create one carbon fragment for biosynthesis of different nucleotides, purines, and finally DNA. And the second role of, as I tell already theoretically, of vitamin B12 is to part in catabolism of odd chain fatty acid and some amino acid, because after the final product of the catabolism is propionyl coenzyme A, which after that is converted to methylmalonyl coenzyme A, which after isomerization is converted to succinyl coenzyme A and can be used for energy production. And the coenzyme of this last reaction, methylmalonyl coenzyme A mutase, is vitamin B12. If we tell about deficiency of this vitamin, it leads to accumulation of methylmalonyl, and methylmalonyl is a toxic for the nerve system. From here, deficiency of this vitamin can lead to, first of all, problem with DNA synthesis, and first of all, in erythropoiesis. From here appear all symptoms of pernicious anemia. From the second part, it's accumulation of methylmalonate, toxic for the nerve system, because it's accumulated first of all, of course, in the blood, after that into the cell membrane, and especially into the cell membrane of the nerve system. And these two symptoms, they are manifestation of the vitamin B12 deficiency. And pernicious anemia have a special, the same marks of it, because here we can see the increasing of the size of erythrocytes, but decreasing of their amount in the blood, and decreasing of hemoglobin concentration in the blood. And usually the main reason of pernicious anemia is the failure of absorption of vitamin B12, and usually with the deficiency of intrinsic factors caused by gastrectomy or atrophy of gastric mucosa. And of course, in this case, we don't intake orally vitamin B12. This deficiency we treated by the intramuscular injection of this vitamin. And the last vitamin for today, vitamin C, ascorbic acid. This vitamin is very interesting because mainly it's present in the fruits and vegetables, citrus fruits, tomatoes, berries, potatoes with the skin, green leafy vegetables, sweet peppers, cabbage also contains vitamin T, many berries, as I tell already. This vitamin is very famous because problems referred to as scurvy deficiency of this vitamin, and is associated with, as I tell already, with the lack of this vitamin, and it had been quite prevalent for centuries, many years. So long ago time we tell about problem of this vitamin deficiency, even we don't know anything about the vitamin. The scurvy kills more cells than they are lost in all SEA battles. But only in 1928 vitamin C was isolated, and its structure was determined only in 1933. Many animals, almost all animals, can synthesize ascorbic acid from glucose. See on the structure of it. Yes. First of all, tell about RDA of this vitamin. Adult, you see, rather high, one of the highest concentration between all vitamins, 75 milligram per day, 75. And during the pregnancy, lactation, and smokers, necessary to increase the dose, daily dose of this vitamin till 100 milligrams per day, 0.1 gram of it. And if we see on the structure, it's like glucose, C, similar to glucose. And as I tell already, many animals can produce ascorbic acid from glucose. Only we and primates cannot synthesize vitamin C due to the lack of one enzyme, 1-gluconolactone oxidase. That's why for us, vitamin C is really vitamin. What is the most important reaction required to the use of ascorbate as a cofactor? First of all, cofactor of dehydroxylation of lysine, and here you can see all very important reaction of this vitamin. First of all, hydroxylation of proline and lysine requires in the collagen. Hydroxyproline and hydroxylysine are essential for the collagen cross-linked and the strength of the fiber. In this way, vitamin C is necessary for maintenance of the normal connective tissue, once healing skin, capillary walls, cartilage, bone, and teeth formation. Vitamin C is required as a cofactor in the reactions of tyrosine catabolism, catecholamines catabolism, synthesis of norepinephrine and epinephrine from tyrosine, because it's a coenzyme which transform coenzyme of dopamine hydroxylase. Vitamin is necessary for biosynthesis of serotonin from tryptophan, synthesis of carnitine, bile acid, steroid hormones, see on this scheme. Vitamin C takes part in oxidation of phenylalanine and tyrosine to homohentizic acid. It protects SH group of protein from its oxidation. It took part in breakdown of histamine, the inflammatory component of many allergic reactions. You see how many function. Ascorbic acid also involved in the non-heme iron absorption in the small intestine, and overall it took part in regulation of iron homeostasis. And one of the very important function of this vitamin, it's as strong water-soluble reducing agent and antioxidant. And the most important antioxidant function of ascorbate is the reduction and reactivation such way of oxidized alpha tocopherol, vitamin E. More detailedly, I will tell you next lecture when we will tell about fat-soluble vitamins. And due to this, it lead to stop of free radical chain reactions on the phospholipids of plasma membrane. Acting as an antioxidant, ascorbic acid itself is oxidized to dehydroxyascorbate. And after that, we can again reactivate dehydroxyascorbate to its reduced form by using glutathione and NADPH. See here that is took part in hydroxylation of proline, and we produce hydroxyproline, and such way we can produce hydroxylysine. Hydroxylation of proline can occur to different positions. We can produce 3-hydroxyproline or 4-hydroxyproline and hydroxylysine. Due to additional of this group, first of all, happen the glycosidation, attachment of carbohydrates to collagen, and formation of additional bonds between collagen fiber. That's why it's very important for the formation of normal, good, stretch collagen. And as I tell already, it's necessary for reactivation of vitamin E, and such way it's act like a very strong natural antioxidant. Deficiency, scurvy, the famous scurvy, the disease that the lack of this vitamin, you see. And here we can see weakness, anemia, swollen tissue, spondygums, open sores of the mouth, losing of the teeth, and first of all, problem with the blood vessels and bleeding. And we can treat it with the adding of fresh vegetables and fruits. Bleeding, you see. And here, you know that vitamin C widely used for the treatment of common cold, and here we used overdose of this vitamin. It's possible, of course, but only for a short time. If we used prolonged time, high doses, very high doses, more than 1, even 8 sometimes grams of this vitamin per day, it leads to toxicity of this vitamin. And first of all, it stimulates formation of oxalate stones in the kidney. That's why it's very dangerous. Okay, that's all for today. We end our lecture and we continue next time. Next time we will tell about fat-soluble vitamins, and I think that we start to tell about hormones. That's all for today. Thank you for your attention. Goodbye.